Well, I was a research assistant last year in my current lab, and I decided that I liked the science and I wanted to keep doing it. So I stayed on at the Doherty as a PhD student. As a scientist, I'm interested in the technologies that we use. So we are interested in the biology as well, but part of science is figuring out what the technologies are and then working out how we can apply that to fine biology. And I'm interested in the technology side. So one of the developments that's happened in the past while is something called single cell sequencing. And what this is about is splitting up tissues. You might have seen in tissue slides where you can take photos of things and see, okay, that's like your muscle, or that's your spleen, or your kidney. Instead of just taking a photo of it, what we're doing in single cell sequencing is actually sequencing all of those cells individually and then seeing what the genetic differences are between those cells. So it's a way of seeing the heterogeneity in a sample that you wouldn't see just from photos. In blood, for example, you've got a mix of different immune cells. If you just sequence your blood entirely, you'll get a big mix of genetic material in there. You don't quite know what it's doing. But when you sequence all of those cells individually, you can actually begin to piece together the network of cells and how they're all interacting together. Maybe there's a small subset here that's only responsive to a certain type of infection, or maybe there's a subset over here that's responsible for, I don't know, dampening an immune response. So all this kind of technology put together and being able to split out that cells this way is really helpful for understanding exactly how our body responds to infection. A day here, I'm a bioinformatician, so a lot of my day is spent in front of the computer. We're coding new software, or we're using existing software to analyze all the huge amounts of data that we have at our disposal. So it's a lot of time at computer, but oftentimes I'll be able to get away from that to have a coffee somewhere next door down the road, have some meetings, or talk to some other lab groups next door about the projects that they're doing as well and collaborate with them.